question to the addition, CACU can be used as a model for the healthy cells. It's not a big problem, but if you look for the expression of some cytokines, CACU2 is not really the best option to study expression of the cytokines by the CACU2 in some specific condition. Well, we had different, obviously different additions, but again, remember, we don't want all the probiotics to have a good additions. We need to know how they adhere, but again, it's not compulsory. And as well, we was checking some markers, because you have some genes that are responsible for the additions. There's a MAP, MOOP, elongation factor, and then some other genes, and easily by the PCR you can detect. Interestingly, most of these genes been described for the Lactobacillus plantarum. But again, we was having even other species here. We had Enterococcus, leuconostoc in these strains. And interestingly, we detected these genes in the other. So now it's coming the question, sometimes when you perform the PCR, you're looking for the specific gene for the specific species that you work for. But sometimes just go a little bit broader. We'll be surprised to find that some genes that are not described in some species could be present. And this is an interesting point to observe. By the way, this work is done with one of my collaborators in South Africa, Kamini Ramayak. So the name is showing what's the origin, Indian. And she was very, very nice girl, because always in the perfect mood, never been in the bad mood. Even if it's happened the worst thing in the life, she still will be in the smile and try to be a positive and find a way. Because we was struggling a lot when we needed to do the RNA work with these strains. To isolate the RNA, to do the expression of these genes, not always working well, but we managed to, because she was studying the expression of these addition genes in Lactobacillus plantarum 423, the strain, to see how the strain can be really acting as a probiotic. But then we decide, okay, let's implement all this work that you already done with plantarum to the other strains. And it was showing that the genes was present, and they was actually expressing them, not only present, but that was actively expressing. And depends on the condition that you have the strain, you can have modulation of this expression. In this case, it's very important when we do the real-time PCR to check the expression on the RNA level when you have the strain in the condition when it's supposed to perform his duty. Because very often you can be really surprised that the strain expressing some beneficial metabolites only when it's in the gastrointestinal condition. In the normal MRS, they don't want to express, but when they go to the gut condition, we can modulate this and we can simulate, you can detect this production. Professor Kivari, can I have a little bit more example and then we have a break? Okay. Then we was using this boza, remember this, I was giving you this example before, as a vector for delivery of probiotic lactic bacteria to the host. In this project, we already know that the strain Enterococcus multi producing antibacterial and antiviral peptides. We know that having a very strong activity against different pathogens, we know that the strain is a very good probiotic, because we even made the experiments in the animal. And very interesting, in one day the Kamini come and say, but listen, we feed our mice with this strain, but you know they're not getting fat, they just build on muscle. Because it's stimulating even muscle production. I say, okay, but why not suggest this to the farmers that do the pig farming, and they can pig with more muscle, not with the fat, because when you buy the meat, you don't want the fat, you want the meat. And then there was actually a different project going as supplements, probiotic supplements for the farming animals, with the idea to build more meat and no fat. So there was even showing pro-inflammatory, anti-inflammatory properties when the animals was receiving this strain, improving the immune response, improving even fertility in the female animals. So it's really complex work, showing the beneficial properties in this strain. Strain was having a very interesting activity against some gram-negative Klebsiella. Some strains being sensitive to the strains, some Escherichia coli, some Clostridium, some MRSA Staphylococcus aureus not showing sensitivity, we checked just 2. Proteus mirabilis, Pseudomonas, not all. We checked 19, 6 only was sensitive to this bacteriocin. But again, this bacteriocin was very potent, and as well the strain applied as a probiotic was having a very good idea. Strain was having a reasonably good growth when exposed to the different percentage of ox bile, and this is important because the strain can survive in our gastrointestinal. And as well in different pH was having a good growth. Well, enterococci normally they don't complain. They can grow in the pH in ox bile. It's a part of their nature. If you have enterococcus that can be fully killed by ox bile is something new. You're going to have a good paper in the good journal because enterococci normally they survive ox bile. But the opposite will be maybe new species. So we wanted to see aggregation property of this strain with a different beneficial pathogen strains. So we have some beneficial, some pathogen strains in this work, and it was showing aggregation because remember, bacteriocin needs to get together with the pathogen in order to kill it. If they cannot attach, it will be very difficult to kill. So aggregation property is a very important preliminary test in order to know if the strain will have a good behavior and good application as probiotics. As well, we was checking this absorption of the bacteriocins to the different organisms, to positive, sensitive and resistant to the bacteriocin, to predict if some specific pathogens will be more or less sensitive. Do you have curiosity how this test is going to be done? Do you want me shortly to explain now, or you want it on the discussion now? Okay, let's do it now. You having a cell suspension of the test organism that you want to explore. You having your cell-free supernatant of your bacteriocin. So you just need to know how many arbitrary units do you have of your cell-free supernatant. How much is the bacteriocin strong there? Then you mix bacteriocin together with the cell suspension of the test microorganism. You expose them, let's say, for 1 hour. You centrifuge, you need to correct the pH again, and then you do again arbitrary units. And you compare arbitrary units in the beginning, arbitrary units in the end, and by formula that is on the papers already published several times, you can calculate this percentage. Very important tip, if you do this test, remember, if your bacteriocin is very strong, you need to dilute. Because something like 12,000 units is a good level. 6,000, 12,000 is a good level. Because if you have a very high activity like 100,000 or 50,000, you're not going to see the difference. Because some bacteriocin will be absorbed on the surface, but compared with the leftover, you cannot see the difference. So you need to dilute your bacteriocin to like 12,000 units, and then you can do the test and you can see this difference in calculating the. We wanted as well to see how the temperature interferes with this absorption. Because look here, you have absorption to Listeria, to Enterococcus fecalis. A very good absorption is in the focus 25 to 40 to 37, more or less here. Less around. So it means the strain having a good potential to be applied in the human being, in 37. Or even in some fishes, because, but if you want to apply this bacteriocin because of his absorption, because you're applying the strain, but strain will produce the bacteriocin. Obviously, in the cold storage food products, not going to work such a good like in the human. So from this point of view, this simple method to do, you do the absorption study at different temperature, and in different pH, you see in neutral, it's working much more better. So probiotic will work much more better in lower part of our gastrointestinal part. Obviously in the stomach, no way. Not going to work very well. Most probably against Helicobacter pylori, it's not going to have a good chance. But down in the lower part of the gastrointestinal part, when the pH is like 6, 7, we'll have the good option to perform well. Different chemicals, we check the absorption in the presence of different chemicals. Some of them are clearly from academic point of interest, some of them from the practical point of interest. But again, you can think about, okay, I have the medication together with our probiotic. There are some chemicals there. I can have the food products and enrich my gut environment with some specific protein, dairy protein or plant protein, and you can actually explore and see if this will give some interference on the absorption process. There was having a very good bacteriocin himself, very good activity, healing very well enterococcus, less little bit recovery of Listeria innocua. But again, this is some points that again will give you indication if can be a good option to cure some of the foodborne pathogens. As well, when we was exploring the percentage of the cell lysis, you see even 20 units and 12,000 units, that was reasonably equal. So you don't need to overload with the bacteriocin. And it's another important point. When you apply the bacteriocin, do you think they will produce in the same way in our gut like in the MRS? No. MRS is a perfect media, and very often they can express very well in the perfect media and much more reduced level when they in our gut. But there was several works showing that in fact several bacteriocins can really express in the gut environment. There was a work showing and proving this. So, but again, even 200 units, that is very low activity. It's enough to perform well against Listeria. Going to a different way, we was showing by the different approaches that we can use the reading of 260 and 420. Remember, here was a DNA, RNA proteins, liberation from the cells, presence of the beta-galactosidase as a fact that it's an intracellular enzyme, and if you treat the cells with the bacteriocin, you destroy them and you liberate all these proteins. We're showing that in fact this bacteriocin was doing well. But there is coming question again, antibiotics. We needed to check. You see a long list of antibiotics, but most of them are from the same groups. And then we was having, including the different antibiotics as well, to be sure about the safety. But again, as I say, some of the antibiotics, we believe that some moderate resistance could be actually a good point. But coming a different point, what about the other medications? Normally, if you are in the perfect health, you're not going to take a probiotics, right? If you start having a problems, you're looking for them. And very often you take another medicines, and especially we need to pay attention to the medicine that the people taking for the rest of their life. If you have, let's say, diabetic, you have the cardiovascular diseases, you need to take this medicine every single morning, no option. And very often these medicines, these pharmaceutical products, they can interfere with the probiotics. There's another point that merits more attention because medical doctors very often don't telling you this. And as we discussed in the beginning, no one read this huge paper inside the box. If you're taking, let's say, ibuprofen, it's an anti-inflammatory. Ibuprofen, it's a group of the sodium or potassium diclofenac. I mean, it's quite popular medicine in anti-inflammatory, but the problem, they have negative effect on the probiotics, they're killing probiotics. If you read this paper inside the pharmaceutical product, it's written, don't take it for too long because it can have negative effect on your gastrointestinal microbiota, but again, people don't read this. This is important. You need to educate the people and explain. Please read it. I mean, it will take you a few minutes to read this, and then you know for some possible negative effects. But several probiotics could be killed by this anti-inflammatory. Some other medications, like even the lactic trioridazine or dioritidine, that is cardiovascular, they was having this killing effect against probiotics. And now you need to do the minimum inhibition concentration, because only by this way you can see if they will have the really negative effect or not. Because if the minimum inhibition concentration is very low, so you need a little bit to kill the probiotic, then you're in trouble. Because You will take this medication like every, say, 8 hours, every 12 hours, once per day, and normally you're reaching quite a high level in your blood plasma of this. And since it's in the blood plasma, this can be transmitted to other environment in the body. And then imagine if you have at this level, high level in the blood plasma, how much is in your gut, even more. So this is important to be sure that medication that you take is not going to interfere with the probiotics. Again, most research is needed in this area because not sufficient information. And even I was having Kiva in there to generate a website, free website, where we can upload all this data by anyone in the world that was doing this kind of work, so the medical doctors can have this access, because sometimes they don't know. They don't know if this interfere or not, but something that merits additional attention. So it's more or less like that. Imagine you're taking every 8 hours your medication. Your level of this medication will go in this way more or less in your body with some fluctuation. And if the minimum inhibition concentration that will kill your probiotic is, let's say, 75 units, obviously you have much more of your probiotic, of your medication in your stomach. This probiotic is not going to work. You will be killed. If you need, let's say, 200 and 150 units of your medication to kill the probiotic, I mean, the level is always like that, so this probiotic will be happy to survive again with medication. For this B, we don't know, because it's fluctuation around, so most probably could be a problem as well. Then we wanted as well to see if we can produce and kill inside the product. So we made it fermented milk, mixed culture of our producer and our test organism. And we was doing a level of pH, how the pH decrease, how the blue is a total cell count. This one is the listeria cell count, and the level of bacteriocin that we can detect. Obviously, we detected much more low activity, only 50,000 units of our bacteriocin in this condition, but as well compared with this is compared with the MRS, but again it was enough to reduce the level of listeria in this system. But then remember, I was telling you that addition of the probiotics can depend of the to the cell line can depend on several components. So we was doing addition to the CACO2 cells, but now taking consideration normal CACO2 cells with our probiotic, in presence of periodate, trypsin, pronase, pepsin, because different proteolytic enzymes will be present in our gut. We was doing all this process in presence of oxbile and pancreatine. You see, addition was very bad when oxbile was presented. So the presence of oxbile can actually reduce drastically addition of our probiotic to the CACO2, but some other components was not really interfering significantly with this idea. And this we can go in the next after the break, because I'm sure you want to have already tea and snack. And since you survived till end, chocolates. Okay, how are we going to do it?
After the lecture.
After? I was thinking no. On the tea break. Okay. Where's the tea break? But where? Okay, let's start here with the chocolate. Can you help me, please? Okay, enjoy. No, it's good.
फोटो ले लेना है।
I think that's in there. Okay. Different. Thank you. More. Okay, let's, now it's working, right. Okay, why is so dark the screen? Lamp dimming. Okay, but there's something we can do on the computer or not? We continue with the story with again. Remember I was showing you this example in the previous lecture, but now we're looking from a different perspective. Now we don't want to kill them. We don't want to kill the test organism. We don't looking for the food safety. You see, the same experiments can be applied in the food safety, could be applied as a probiotics experiment. Because we're producing boza, we're making all this process, we inoculate, remember, with the boza, with the test organism. We wanted to know what is going there. They're growing quite acceptably. You have the good population numbers of the microorganisms. And then you're having the DG. You want to see, in this case, I don't care about disappearing of Lactobacillus acti because it's not my objective to have disappearing of the test microorganism. My objective in this case is to have Enterococcus multi present in the system. And the strain is present because it was confirmed by the normal species specific PCR, and this will be where is here. This PCR confirming that Enterococcus multi with the species-specific primer is present in the product. So starter culture Enterococcus multi, making the Enterococcus multi to be present in the process. And in this way, we guarantee that our product, fermented food products, can carry the probiotic strain. Again, analysis of the organoleptic properties was clearly showing that the consumers cannot detect the presence of the extra strain. And this is extremely important when we develop the new fermented food products, because if you change the organoleptic properties, people don't buy it. They don't want it to have it. And you need to make them happy in order to provide these strains and these products to them. And he was using actually GIT model. Well, most probably in the literature you saw already Scheim, Team, etc. We have this model in South Africa we call Frankenschwein. In German, Schwein is pig, Frankenstein, Frankenpig. And interestingly, it was easily working. You can see you can easily make this gastrointestinal model in your lab. You need a chamber to put all these things because it needs to be an appropriate temperature. You need some pumps. You move from one another part of the stomach. You add in some gastrointestinal fluids. You can take a sample. You have the pH meter inside to change to correct the pH, etc. And you can do it relatively easy. It's a dynamic model. However, it's very simple, but again, it's working. Now we're developing the new model in our laboratory, then we can actually easily follow the changes in the microbiota. We're going to inoculate our models with appropriate gastrointestinal microbiota. But the important thing, we need to pay attention, because imagine if you have milk here, it's okay. It's working perfect. But imagine if you put the grains, if you put the corn going to go via these pumps? No way. So we actually modifying our pumps in order that even the pieces of the food can go via the system and could be applied. And hope in the next couple of months we'll be finalizing and already running our first experiments. This is together with our collaborator in Russia and doing this kind of work. Even there's some models now, they actually was linked together with the sequencer. So automatically they can take material from the model, they can do even genetic sequence, they can do metabolomic, everything automatic. But where's the fun? I don't want a model that you put something in the front and you wait 1 week to have the results. I want to have my hands there and to know, to have the control on this process, to enjoy all this work. We have another kimchi project, that's called kimchi project, where we was exploring the different probiotic properties of kimchi. In this kind of work, we wanted to know, to develop actually some alternatives. So we was looking for the appropriate strains in order to select the good strains for the developing of new therapeutic products. If you look here, the antibiotic discovery resistance timeline, when the antibiotic being discovered already 12 years later, you had resistance. Then you have more antibiotics and again resistance. These days we can have some alternative. We can have several bacteriocins listed here, and they can be applied in a different process for resistance to different diseases. I mean, we can have the area of application, vancomycin resistant, insulin resistant Staphylococcus aureus. We already have application of niacin and lactacin for treatment of some MRSA or vancomycin resistance. In case of treatment of Clostridium difficile, we have multicin, niacin as well applied, but generally multicin. All these dental care, contraceptive, skin care, tuberculosis, antiviral, antifungal, different pharmaceuticals, different bacteriocins with different applications. So it's showing that in fact bacteriocins can be considered as a next stage to kind of, at least temporary, win this battle against antibiotic resistance, because we needed more and more way to do this kind of treatment. In this kimchi project, actually we was isolating different microorganisms from the kimchi. You see how they're producing kimchi. They need to treat, you know already the recipe, or you just know the product? Okay. They needed to incorporate these spices, red chili and salt and everything, but again, we was trying to isolate microorganisms, and actually it was quite a lot of microbes there. It's a really living system. They're having jokes. Sometimes the students go home, and they're going for the kind of holidays, whatever. They're coming back after that, and they taking kimchi with them back from home to the university. Very often they carry kimchi in the plastic bag, and the kimchi is fermenting even in the tray. And sometimes bag can break and you can smell it very well even in the tray that someone carrying kimchi. So don't be so positively about the kimchi. Most of the kimchi have a very good taste, rich of several beneficial microorganisms. But since the kimchi is a cabbage, right? And the cabbage is growing where? On the ground, no way. And very often can be contamination with bacillus. Even we detected in some samples, even Bacillus cereus. And Bacillus cereus can be a very nasty foodborne pathogen. So, in fact, very important hygienic condition in the production of the kimchi needs to be applied. And you can believe me, not always people manipulating work like that. Sometimes no appropriate clothes, no gloves, no even appropriate containers to do. So it's important to have the appropriate hygienic conditions. So isolated some of the bacteria, we have production of the bacteriocins, and then we was doing a REP PCR to prove that we have the different strains. And we have two different strains, Enterococcus faecium and Enterococcus 120 Kc. Well, you see ST my initials, 20 number of the isolates, Kc kimchi. And then we had isolates 41. And these two strains was showing very good survival in the part of the gastrointestinal condition. So when they was passing the gastrointestinal condition, you have the time 0, time 1, time 2. Time 0 is like what's going in the mouth. Time 1 is considered when they stay in the stomach. And in the large intestinal it's time 2. So you see in the case of strain 20 Kc, the first strain, you're having decline, but still, okay, look, it's decline, but this is 8.10 to 8, 4.10 to 8. So sometimes you need to see very carefully the scale, because it's declining just even not one log. So it's actually, it's a good survival. It's, again, if you look here, it's a look big, but you need to look the scale on the side, what is it? And at the same time, Enterococcus fecalis, a probiotic strain from the laboratory, was having more or less the same same behavior. So the strain was having a good potential, these two strains to be applied. And then we wanted to see how, when we do the combined culture, because not only to have the probiotic strain, but we wanted to see when the probiotic strain is co-culture with the Listeria monocytogenes, because we know the strain having a negative effect, inhibitory effect against Listeria. But in this case, we culture together, bacteriocin producer together with the Listeria. We see the good survival of the bacteriocin producer, but practically for almost 100% remove of Listeria from the sample, from the environment. So if you have the patient that having Listeria contamination and taking this probiotic, in fact will help to not only to remove Listeria but prevent of future Listeria contaminations. For the people that are in the high risk, in fact this kind of probiotic will be perfect because will be helping in prevention of the Listeria contamination. Well, the best thing, if you are pregnant, please don't eat the food that is a high risk. Because Listeria monocytogenes could be killing not only your future child, but can kill yourself as well. Don't give it to small children, don't give it to the elderly people, don't give to the people with the immune response problem. Now, when you're talking about immunocompromise, what is coming in your head as the first option? Which people are immunocompromised? Come on, answer. Can I put in the test this question? Well, normally the people always will say HIV. They're immunocompromised. But they're just a small part of the immunocompromised people. If you have organ transplant, you're immunocompromised because after the transplantation of the organ, you need to take for quite a long time immunocompromised medications. If you're in some chirurgy, you're in the stress, you're immunocompromised. For all these people are actually as well, they're immunocompromised. Even as I say, you're making exam, your immune system is going down. So all the pathogens will take advantage and try to make your life more complicated. So we made it as well the metabolomic analysis because we wanted to see what kind of metabolites are produced by these strains, because obviously not only bacteriocins, and unfortunately it's not in the color, but darkest color normally they have more present. So you see several acids was increasing in these strain environments. So not only bacteriocin but the acids and even some additional antimicrobial metabolites detected, and that was improving the antimicrobial effect of when these two strains was growing. So in this case, effect of different enterocins produced by different strains of Enterococcus faecium, obviously they have this very strong effect. And we was actually selecting for future study a different strain, Enterococcus faecium 7119, isolated from, this is the initials from one fermented soybean pest in Korea. And we wanted to see again how they would. So we treat the vancomycin-resistant Enterococcus faecium with our bacteriocin during the flow cytometry to follow what's going. You see have live, damage, and dead cells. In most of the cases, this is the control, and different level of the bacteriocin when applying. You have a lot of dead cells and a very small amount of damaged cells when you apply 12,800. When you go further, you have kind of breaking point between 6,000 and 3,000 units. So now we have live cells, dead and damaged cells. So obviously to be effective this treatment, you need to have something a little bit more than 3,200 units. Because if you apply less, most probably you will actually not kill. You can have several damaged cells, and these damaged cells actually later could be maybe recover and make you problem. If you use much less bacteriocin, you have several live cells and a little bit of dead cells. So minimum inhibition concentration is somewhere here between 3,000 and 6,000, and you need to apply it again in enough concentration in order to be sure that you're going to reduce and you eliminate, not just to making sleep this vancomycin system, but really kill it and remove it from the system. So we was using, again, to explore the beneficial properties of symbiotics between the bacteriocins and some antibiotics. In this case, we was working against vancomycin resistant, to kill the vancomycin resistant enterococci, and we was applying combination of our bacteriocin and vancomycin. So you can see in these two cases with the two different. This is bacteriocin 1, bacteriocin 2, bacteriocin 1, bacteriocin 2. We was showing that in fact when we apply the vancomycin, they're not supposed to work because they're resistant. But you apply it together with the bacteriocin, they work in combination. And even you can reduce the quantity of antibiotic, and antibiotic that is already been a problem, it's not working because of resistance of the test microorganism. Together with the bacteriocin, you can have the effect and you can have the beneficial properties. As well, the ciprofloxacin, use it as a control. We was observing the same pattern and again interaction between bacteriocin. Again, doing the same experiment with the live dead cells, we was observing that in most of the cases dead cells and not live, not just damage, and effectively working. Then we wanted to analyze this data, and we was performing, because we're having kind of mathematical models that can tell if it's a synergistic, is it just the additive effect, indifference, or antagonism. Depends on the level of this factor, 0.05, 0.5 to 1, below 2, or between 2 and 4. And when I was performing these experiments, in fact, different concentration of the antibiotic and the bacteriocin, these indexes, in fact, was showing that in most of the cases we was having synergistic effect. So it was confirming that we can apply the allobacteriocin together with okay, it's almost 10 to 12. We was using fish flow application as another, and then after finishing with this example, we can actually go for the lunch break. We was exploring the use of fluorescent in situ hybridization together with flow cytometry. We wanted to combine this method because we was having this idea. Normally the people do a fluorescent in fish application when you use the specific fluorescent marker that they will attach to the cells, and based on the fluorescence you can know what do you have. Oh well, but this image was telling you something. It's telling you, yeah, I have live, I have dead cells, I have cells that I'm targeting, but how much? It's a little bit kind of, okay, it's a nice image, but not really giving you a significance of this treatment, because you're having the cells, but they are too much, they are less, they are acceptable, they are non-acceptable. And actually, the idea is to combine the fluorescent in situ together with the flow cytometry, and then you can make a quantification. How the process more or less works? You need to select and design your probes. So you need to have specific probe that will give the fluorescence. And I just, please, the girls from the lab of Santos, remind me to generate PDF today on the lunchtime and to put it on the system. Because I always forgot. Can I do? I can do it in this computer in the end. I think it's easier, even. So then you design your probe. It's not a really big problem. This have several computer programs these days. It's quite easy. Then you need to select the way of the samples, and you need to perform first hybridization. So you need to find appropriate conditions, because you play with the temperature, you play with the time. So to have a really targeting, specific targeting of your probe with the DNA of your organisms. So you have the samples, you make fixing, you put your marker, and then after that, doing the hybridization, you need to do visualization. You can do it with microscope, but again, it's a little bit complicated. So after the fixed and permeabilized cells, you're having this process of the putting your target, your marker where you want. You made it a contact with between the ribosomes and your marker, and you can observe. In this case, we're going a little bit further. So we're doing the samples, we're preparing, we redesign, we're doing in the appropriate concentration in order to work. And then we put our fluorescent marker, doing incubation and then going to, okay. Well, I was supposed to click to go to those steps. And then you're going to the flow cytometry when actually you can analyze. In this work, we were taking to want to validate our idea because we made it in the pure culture. So we took the different samples from the patients from bacterial infections. Here we do the classical microbiology to see what we have there, what we don't have. But then we was performing the analysis, and we selected appropriate vancomycin resistant strains. We was isolating the strains that are purely vancomycin. So we wanted to detect vancomycin A and vancomycin B genes. By the plate, we know that our bacteriocin is working against this vancomycin resistance. And when we do the fish flow cytometry, in fact, we was able, okay, we was able to detect much more quicker by flow cytometry, because if you do the isolation, if you do the test, you do the antibiotic resistance, you try to kill, it's taking a long time. But with the flow cytometry method, you have the marker for the vancomycin, you have the marker for the vancomycin B, so you actually make the probe, they detect, they catch these genes, and with the flow cytometry you can analyze. So in this case, our strain, Pediococcus pentosa cells, bacteriocin producer, was able to inhibit vancomycin resistance and to able to kill it. So with a very small amount, we was able to perform this work. We was showing even interaction but when we was doing flow cytometry, we was able to detect the fact of the presence of the vancomycin resistant genes in this total isolate. So if you work in the classical microbiology, you isolating a strain, you trace this strain. If you do the fish flow, actually you can take a total sample, you have the probe that you already validated, they recognize the vancomycin gene, they was making contact, they do the fluorescence, and by flow cytometry you can analyze and you can say, yes, our sample is carrying vancomycin resistant genes. And obviously you're not supposed to use the vancomycin for treatment of these patients. So we get even further. We developed the probes for the